Hi there, Year 12. So we're down at Millook today, north coast of Cornwall. There's lots we can do down here related to the spec. With Module 4, there's all sorts of stuff. You've been looking at some classification and also some fieldwork techniques. And then in Year 13, with Module 6, it all overlaps there, so we can tie Year 12 and Year 13 together whilst doing some fieldwork down at Millook.
Also, just want to say, those Year 11s that are coming into the sixth form, this would be a great introduction for your biology A level to see what's expected. So have a watch of it too, because you'll be doing this very quickly in the new year. Is that right, Mr. Ryan?
Absolutely.
Absolutely. Right.
So before undertaking any field work, we have to obviously do lots of risk assessments. There's all sorts of risks on a rocky shore, from slips, trips, falls, as Mr. White has just demonstrated. But all of this has been risk assessed, and whenever we do field work it would always be risk assessed. It's coming up to low tide, so we've got an outgoing tide, and we'll start at the low tide mark and work our way back up when we actually start sampling. So the first aspect that we're going to look at is diversity, and diversity, you might be familiar from your spec. We can split that into three straight away. So we could have habitat diversity, as mentioned. This is a rocky shore, but it could be a sandy shore like a Widmouth or Summerlees, could be a woodland, could be any ecosystem. You can also have species diversity. Obviously that's not only how many different types of species there are, but also the relative abundance of each species relative to the other species, what we call species evenness. And also you can have genetic diversity, so the polymorphism, the variation within the species or within a population today. So when we come to sampling, we can divide sampling into several different categories. We could have opportunistic sampling. That's the easiest sort of sampling to do, but probably the least effective. That would be, for an example, you could go to the sixth form common room and sample the students that were there just because they happen to be there. But maybe they're not very representative of a population of students. They're just the ones that happen to be there, so it's for ease. So we're not doing that today. You could have stratified sampling, where you split your sample into different strata. Could be males and females. And what we're going to do today is systematic sampling. And our systematic sampling is going to be non-random. So for non-random systematic sampling, we'll be looking at a line and we'll be sampling along that line to see how populations change with distance from the low tide mark. So if we were random sampling today, what we would want to do is grid out the entire area that we're sampling. We could use Google Maps, we could use whatever, or we could just use a tape measure and lay it out. We would then generate random numbers by a random number generator or rolling the dice or any other random means, and then we'd use a quadrat, which is essentially just a square that would be placed at random according to coordinates that have been generated randomly, and we would see what species we find within it. So a quadrat's just a square. This one's 50 centimetres by 50 centimetres. Which is the average size, but you can get smaller ones, you can get huge ones, you can make them yourself. They can be made of string and rope, so they're more portable. And possibly more useful is this grid quadrat, which is split into 25 squares. So that means each square is 4%, so we can very easily and quite accurately estimate percentage cover of certain species. Today though, we're not going to be doing random sampling, we're going to be following a transect. So for a transect we need a line, and for the line we'd lay out a washing line. Because we're in a big environment, that washing line won't be long enough, so we'll just keep moving it, but we'll follow a straight line up to the high tide mark from the low tide mark, and we would have several repeats along the shore. So we'd maybe have 3 transects. And what's interesting about a rocky shore is it's not the horizontal distance that is important. What dictates what lives where is the vertical height above the low water mark, because that dictates how long you're submerged or above water. So for that we can have a vertical transect, and we'll use what I would call height sticks.
Geography we call them ranging poles. I think that's the proper term, Mr. Wright.
And like that we can very easily have one station where we have a quadrat, and then the next quadrat will be 1 metre higher up. And then we can have stations moving up the shore until we've sampled the whole vertical profile of the shore. So one of the hardest things about sampling on a rocky shore is being able to identify the species. Obviously there's keys available and guides and seashore guides, lots of which are fantastic, but seaweed has a horrible habit of looking like what it's not. It looks completely different when it's dry, it's completely different when it's wet, looks different based on its age, looks different based on how exposed the shore is. So we'll have a look at some identification that we can do that we'd call indicator species. So this green weed at the top of the shore, we'd always expect to find green weed. This, people like to think, looks like guts. It does under the water. If we pick it, this is Entramorpha intestinalis. We can see it's a green seaweed, it's a photosynthetic seaweed, but all seaweeds are photosynthetic. They just use different pigments which determines their colour. So this is Entramorpha intestinalis, that's the species we'd be looking for at the top of the shore, and we've got it growing there. But some of the others we'd expect to see, if we start way down at the bottom of the shore, subtidal, which is why we can't see it. It's underwater. It's not a huge tide today. The big spring tides where the sun and the moon are pulling together have just gone. We've got a medium-sized tide today, and all this kelp, which is a brown seaweed, is buried underwater. So this might be up to 20 feet long, reaching up to the surface, flowing around so that it can photosynthesize. There's lots of different types of kelp, and I just found these on the tide line. But the structure is all very similar. It'll have a holdfast where it attaches to the rock or to the strata. Then they will have a stem, which we call a stipe, and then the fronds, which flow off beyond that. And obviously when it's flat, it doesn't look very nice, dried up on the tide line. But underwater, when it's all flowing, it can be beautiful and huge range of colours. So that's the lowest end of the shore. As we move up, we've got this weed, which might look very like lots of other weeds, but we can see that it's quite conspicuous in that it doesn't have air bladders. Still got the stipe, would have had a holdfast. It's got a midrib up the middle. It's got these fruiting bodies on the end, which are actually the sexual organs full of gametes and jelly that they use in agar and makeup and all sorts of things. But there's no air bladders. And now it's dried out, it's almost kind of twisting. And that's why this is Fucus spiralis, it's spirals. Then we've got one that may look very similar, but we can see there are actually a couple of air bladders. And usually these would be in pairs, still got the fruiting bodies, but the fact we've got these paired air bladders either side of the midrib, that means this is Fucus vesiculosus, or bladder rack. We've got a flat periwinkle feeding on it. It might look really conspicuous and not a good idea to be bright yellow, but maybe you can think about why it might be bright yellow, because there are selective advantages to being bright yellow. And this is the species that shows huge genetic diversity, polymorphisms, from really yellow to orange to olivey green to dark brown to almost black, and every shade in between. And they all compete successfully, but for slightly different reasons. Finally, we've got this one, Ascophyllum nodosum. Here, huge air bladders, and they're right in the middle of the frond. They're not either side of a midrib. There isn't, in fact, a midrib on this one. And these air bladders, different to the fruiting bodies, are full of gas to enable the seaweed to float so that it can access the sunlight for photosynthesis. So here we've got our different seaweeds. We've actually only looked at brown and green so far. We've actually got red seaweeds to go, but we'll stick with these for a minute. So the structure is pretty much the holdfast, which grips to the rock. It's an incredibly strong glue that they use to adhere to the rock. Then we've got this stipe, which is the stem for all intents and purposes. And it always splits into two, so what we'd call a dichotomous split, because it splits into two, like so. And on the ends of those, it might split again. We've got those fruiting bodies, but there's this midrib that we can see usually on lots of species, Fucus vesiculosus especially, and we get the bladders either side. the air bladders either side like that, which we can see here. So these air bladders either side of this central midrib. And if that's the case, then it's fucus vesiculosus we're talking about. But we've got a holdfast, we've got a stipe, we've got a midrib, and we've got these paired air bladders. And possibly we've got these fruiting bodies on the top, full of jelly and the gametes, which will help these to reproduce sexually. So there's a real load of jelly in there, really smooth, slippery stuff. So the next thing we're going to look at is some of the shells. Again, we're just looking for species that we can easily learn to identify, and that will tell us something about the environment we're in. So when we're looking at shells, got a little selection here that I've all just picked up off high tide line today. So these are all dead, they're empty shells, but they help us identify the living specimens. So to start with, we want to turn them over. And when we turn it over, we'll see one of two things. We will either see a circular operculum, which will mean it's a top shell, which these have, or we'll see a teardrop shaped operculum, and that's our first distinction. So this would be a winkle of some kind, mostly they're periwinkles, and the round ones will be a top shell. Then if we look closer at the circle, if you look at the operculum of this one, might be able to see a hole right in the centre. So it's called an umbilicus, but there is, if we look at the whorl on the bottom, we've got a shell shape like that with a circle there, and there is a hole, which is the umbilicus. And that will tell us that it's a gibular shell, a top shell, and it will either have purple marks or it'll have grey marks. These ones are all purple top shells, they're slightly smaller, but it could be a grey top shell. Same genus, gibular, different species. Then we've got these, which are periwinkles, no hole, so we know they're top shells, but they've also got no spire, it's flat on the surface. These are the ones we mentioned before that have all this range of different colours, and much darker ones too. And the reason for that is when they're underwater on seaweed is beautiful, bright, yellowy, green, translucent sort of colours, and these yellows are incredibly camouflaged. But when you're further up the shore and you're exposed to the air and the seaweed's all dark and brown, it's much better advantage to be dark. So when you're underwater, you're less likely to be predated upon if you're yellow. When you're out of the water, you're less likely to be predated on when you're brown. So you might come up with a hypothesis when you're sampling of where would you find which colours on the shore, and if it's a sensible hypothesis, you'll be right. This one is different again. This is a predatory whelk, this is a dog whelk, Nucilla lapillus, and we'll see these further down on the shore as well. And these bigger top shells, these are living ones now. I've just picked one straight off a rock. I'll make the snail go back inside. There's no hole. It looks like a periwinkle, like these top shells. Sorry, not a periwinkle, looks like a top shell, but it doesn't have the hole. But what it does have, try and point with this pen, is a tooth. It's just got a little notch in the upper column there, and that tells us that it is a toothed top shell, or Monodonta lineatus would be its binomial nomenclature. And those are the indicator species that we'd look for. We've also got limpets. The limpets are all Patella vulgata, unless we go right down to subtidal, and then you might find some blue-rayed limpets, but we won't see those today. So those are the shells we want to try and be able to identify. So hopefully we now have established what species of seaweed we might be able to identify, and what species of shell. They're going to be our indicator species. Now we're going to go down to the bottom of the shore and start sampling.